Chairman, thank you, ladies and gentlemen. It's a pleasure to be back here. I actually gave up my 4 years as Chair and Vice Chair of the British Society for Genetic Medicine on this stage last week. And I warned our delegates, as I'll warn you, this is a very safe area, but you are in an urban environment. And Frankie Boyle pointed out the local anthem is Never Walk Alone. My small title is How Genetics Will Impact on Healthcare, and I hope I can give you a feel for that over the next 20 minutes or so. This is the one technical slide I promised. I was 17 when our biology teacher took us to hear an Oxford Don explain the genetic code. Everybody's heard of the double helix, but actually for me the most exciting thing was cracking the code. The 4 letters in the RNA, the U, C, A and G, turned into a code for the amino acids in our body, and you have to have sets of 3 in order to get enough of them to cover all the amino acids and some grammar. On the way back to school, our biology teacher asked me to explain it to him again because he hadn't quite got it. For the first time in my life, I had one up on him. So I kind of stuck with me, and when I went to medical school, I decided to take some time out and become a geneticist. Now, I wanted to look at that code because it's really important now that we are conscious of one of the major problems it presents. Look at just one code. The letter C, C, and G gives you a proline. The problem is, if you look above that, a CC in anything gives you a proline. It's called a degenerate code. It means you've got 64 combinations, but you don't need them all. And that presents us with one of the first problems we have when we start looking at our DNA. A lot of the letter changes make no difference whatsoever. That's the first time we got an applause for that line. It's a good start. Anyway, I'm not a DNA wonk. I'm a doctor like you, and I'm actually a family practitioner, as I was saying to Trish. I actually see families over generations, and this is one such family who actually posed for our brochure in our institute. I saw Trudy when she was a little girl and diagnosed that she had Noonan's syndrome. I saw again when she was 20 because she was thinking of having a baby and knew it was a dominant. And we talked it through and she decided to take the chance. And she had Lauren, who is a beautiful little girl but also has Noonan's syndrome and had quite a severe heart defect, which we pointed out to her paediatrician when he'd missed it. The reason we spotted it, of course, because we knew the syndrome and we knew that there was a high risk of a heart defect, and so we went looking for it. So that's an example of the sort of families that we see in the clinic. Rare conditions, 8,000 and more of them now. And the questions we get asked are remarkably similar. In addition to the doctors that you all did, so the questions the doctor gets asked, what's wrong, what's the future hold, what treatment can you offer me? We get asked, why did it happen? Will it happen again? Will it be as bad? Are there any tests? And of course now there are thousands of tests to be done, and that's the challenge that we all face. So when we were investigating Laura and that family, I actually sent DNA all over the world. It was eventually a Japanese team who found a gene called BRAF was mutated in that family. And we used to spend hours sitting in rooms like this, looking at pictures, trying to decide which gene to test. And then one day I actually said something that was sacrilegious to my dysmorphology colleagues. I said, why don't we just shoot the lot? Because we'd actually now got a technology that could actually just look at all the genes that cause Noonan's syndrome. And we've set up a little spin-out business, sorry Trish, from our foundation trust in collaboration with St. George's. And for one price, we can do every gene for Noonan's syndrome. So that's transformed our ability to actually make diagnoses, where instead of spending a fortune and time going through gene by gene, we can now look at the whole set. And indeed, you can now do a whole exome for about £500 and look at 20,000 genes. So isn't that a step forward? Well, it presents certain problems. The first thing is understanding what those mutations mean. So the most important second thing I want you to take away is the idea of conditions which are dominant and recessive. That is to say, the gene for retinoblastoma, it runs in families as a dominant trait. You see it running down from generation to generation, but not everybody who carries the gene gets a retinoblastoma. It's common but not inevitable. And it was actually Al Knudsen who realized what was happening, that it was the two-hit problem. That every one of your embryonic stem cells in your retina, if you have this condition, inherits a faulty copy of the gene. And then it's a chance event whether the second copy gets knocked out. If it doesn't, and you get to the age of 5, you're fine. If you lose that second copy, you will develop a retinoblastoma. And Al very cleverly worked that out from the age of onset curves of this condition. So any of us could get a retinoblastoma, but we need 2 mutations in the same place at the same time. If you have one of these hereditary cancer syndromes, you've already got the first mutation, you only need a second event to get the problem. I always think of bouncers on the door because I come from Newcastle. We have 2 bouncers on the door, but if you have a pub that only has 1 bouncer on the door and he goes for a fag, you're in trouble. So it's a lot easier to get cancer. That works for patients, I find. So having spent the first part of my career being a dysmorphologist and doing fetal medicine and so on, I started to become more and more interested in hereditary cancer because we could predict and prevent cancers and actually prevent ill events. And it was this family more than any that reminded me of this. About 20 years ago, Diane came to see me with her sisters, because their mother had just died of breast cancer, and there had been multiple breast cancers in the family, but also their cousin had just died of a spinal cancer. And that made us wonder about a very rare condition called Leigh-Fraumeni syndrome. And so Jenny Varley across in Manchester started looking at the P53 gene, and we sent all the sisters for a mammogram. Carol had a breast cancer, so they were going to do a mastectomy and radiotherapy, but we said, hang on, this is a gene that makes you radiosensitive. It might be better to, in this case, opt for bilateral mastectomy and reconstruction. And when they did, she found breast cancer in the other breast, which had not been detected by mammography because she was too young. As a result of that, and had we left them to their own devices, all 4 sisters would have all had their breasts removed. But we asked them to hold on, and we in fact managed to identify that there was a gene mutation in this gene, and we could tell that in fact Diane was a carrier, but the 2 other sisters weren't, and therefore the other sisters didn't need mastectomies. And that was a tremendous boost for the family, because at last we were on top of the condition, and we at least saved 2 of the sisters from prophylactic surgery. I was very embarrassed by the newspaper article, partly because of my hairstyle, partly because of the, sorry, I put a ring around, but you can't see it. That's me on there. I got all the credit, even though it was actually Jenny Varley who found the gene. But I was particularly concerned because my mother was still alive, and she used to read the paper, her eyes were going, so she just used to look at the pictures and read the headlines. So, a lot of our business now is tracking down these families with hereditary breast cancer, and it's become about 1/3 of the work that comes through the genetic clinics. And of course, BRCA1 and BRCA2 are the most important and famous of this group. And Alison, I'll remind you, just to sort of take an image away, a rather sad image of Alison. She was a science teacher herself at the age of 50, came to see me in Penrith, and she, at the age of 30, had a breast cancer and had an incredible family history of breast and womb cancer, as she described it, but had been constantly fobbed off in her requests for referral for genetic appointment. And when she eventually got to see us 20 years later, I was convinced of her story, but according to guidelines shouldn't do the test because in fact I couldn't validate her family history. But she was a very good historian. While we were waiting the 6 months for the report 4 years ago, because it was a bit slower then, she was admitted to Carlisle having had a stroke. And she wouldn't let them examine her till they did an ultrasound of her pelvis, and there they found the 2 very large ovarian cancers, one of which had metastasized to her brain. She came over to talk to the medical students when that picture was taken and died about 3 months later. That was a completely unnecessary death. Had we referred her earlier, had we investigated her for BRCA1, BRCA2, we could have found that mutation, taken her ovaries out, which is standard of care now, and avoided her death. So that's the problem that we face. But of course, it's finding the Alisons out there in the tens of thousands of people who have a family history of breast cancer. And of course, that's where we need you to help us to draw that triage line and try to get the right people in, but not reject them inappropriately. And of course, the woman who's done more to teach everybody in the world about genetics than all of my creed or crowd is Angelina Jolie, when she very bravely announced her own decision to have mastectomy because her mother had died, like Alison, of ovarian cancer due to BRCA1. In a moment, she became the best teacher of DNA testing. But it was interesting that nobody said, what's a DNA test? Because in fact, she joined 2 other great teachers about DNA, Monica Lewinsky and O.J. Simpson, who had actually explained to all the people on the planet the power of DNA testing. As a result, though, of Angelina's intervention, we now have people realizing there is a test and they can be told whether or not they're at high risk. And that's going to change even more dramatically next year, thanks to an invention of a chemist in Newcastle about 20 years ago of the PARP inhibitors. And the first of these is about to hit the street. Now, poly-ADP ribose polymerase, to give it its Sunday name, is an enzyme that activates the repair system if you break your DNA. And so single-strand breaks are fixed by PARP and its friends. And the idea of the discovery of the drugs was if you take out that enzyme, it might disadvantage cancer cells. And of course, as you know, oncologists basically try to poison your cancer before they poison you. And so they were looking for something to poison cancers with, and this looked like a good one. But it didn't work, in fact, because there's a side road. If you block the A1M, you can go up the A167. And the same thing applies here. There's a rather winding road called homologous recombination that if you block that top line, you still get there without going down this other route. But if you look down there, you can see there are two purple ones, BRCA1 and BRCA2. So in other words, if a cell is to use that alternative route, it must have a functioning BRCA1 gene and a functioning BRCA2 gene. That is to say, at least one of the copies of the gene must be working. If you've got a cancer in these families, you've obviously lost the second copy. So the cells that are malignant in someone with a BRCA1 mutation will not be able to use that second pathway. So the PARP inhibitor should kill those cells. And the latest evidence suggests that in ovarian cancer, a drug called olaparib does just that. You can see from our point of view collectively as clinicians, we're now going to be faced with families with a suggestive history and someone who's presenting with ovarian cancer. So we're going to have to really get our act together to test for BRCA mutations much more quickly. Another condition which I'm very personally involved in is, it used to be called Mio-Torre syndrome, is now called Lynch syndrome, and is due to a problem with DNA repair also called the mismatch repair genes. And this is probably even more common than BRCA1 and BRCA2, and I want to tell you about it for an important reason you'll see in a moment. I actually came across this family up in Northumberland, and Ted in the middle there was a Berwick farmer who'd had colon cancer 3 times and skin cancer 9 times, and his sister Bertha had endometrial cancer. And in fact, all of the people shaded yellow there had either a bowel cancer or an endometrial cancer. And we were asked by colleagues in yeast geneticists, in fact, in Boston, if we had any DNA from such a family because they had a hunch. And we sent it across, and it turned out to be true, that in fact this family had a mutation in their spell checker. So basically their colon cancers were full of spelling mistakes. Because what if the next slide shows you the idea, these bits of repetitive DNA where you get like 25 T's in a row, incredibly difficult to copy. And so when you're making a new copy of your DNA as your cells divide, it sometimes slips. And remembering back to that first slide, if you stick an extra T in there, everything moves down 1. So you've wrecked the gene from then on because everything is a different amino acid. So you've got to be sure that those bits of so-called microsatellite are accurately replicated. And these genes act like a sort of hand that runs down behind the polymerase, making sure that it's exactly matching the old strand. And if 2 T's come together, they won't stick. So you get a bump, and that bit gets stripped off and run again. So if your mismatch repair genes, which work as a team, aren't working properly, then you haven't got a spell checker. You can still type on your word program, but there's nobody coming along behind saying, whoop, you just spelt that word wrong. And so there's a huge increase in mistakes in the DNA, and that's what we can detect. And 2 years ago, I did my major claim to fame, having 20 years spent 20 years collecting 1,000 people in 16 countries to give them aspirin, because my old friend Gabriel Kuhn in Melbourne had pointed out that people on aspirin seem to get less cancer. So we found 1,000 people and randomized them blind to take 2 aspirins a day for 2 years, and at 5 years, their cancer rates were down by 50%. So finding these patients is now an imperative because we can actually reduce their cancer risk dramatically with the cheapest of treatments. Interestingly, of course, the latest follow-up on the Old Women's Health Study has shown a similar but smaller effect of low-dose aspirin in the general population. So essentially anyone over 50, especially with a strong family history, could justify taking aspirin. But if you've got the gene for Lynch syndrome, you certainly should be on aspirin. And as of this week, you should be on our new clinical trial, which we, after much thought, we'd call CAP3. And CAP3 is a dose non-inferiority study. We're looking for 3,000 gene carriers, and we'll give them either 600, 300, or 100 milligrams of enteric-coated aspirin, and that keeps me in work for another 7 years, and they can't sack me. But even before that, the evidence for aspirin is really quite dramatic. This is unpublished data from last year. So of the 427 people who were on aspirin in our study, and remember they took aspirin for 2 years, then stopped, and there were 25 bowel cancers. In the 434 who were taking the placebo tablets, there were 45 bowel cancers. So you can see that if we can offer a clear clinical utility, as this is an example of, then genetic testing is obviously defensible. And it's becoming dramatically easier. These are 2 of the 4 teams that did the Human Genome Project in the 1990s. All of their production from 1 month can now be replicated by one of our technicians with one of those machines. In 2005, there was a breakthrough in DNA sequencing, and massively parallel sequencing was invented. And there are now a cluster of technologies which have dramatically changed our ability to do this. So whereas in 2000, when our Centre for Life was built to celebrate this whole process, we thought it would cost about $100 million to do a whole genome. When Mylene Creek, one of the clinical geneticists in Leiden, was sequenced by Johan Den Dunnen, it cost him 45,000 euros. And in fact, the Americans were a bit sniffy because they'd already done Craig Venter and Jim Watson. But the Australian newspapers got the tone of the day when they said, man takes one step towards understanding woman. Very small step indeed. So we're now in a new era. And in fact, a couple of weeks ago, I was privileged to attend the signing ceremony at 10 Downing Street when Jay Flatley from Illumina, who made the machine I just showed you, which is a British invention, I would add, is actually going to build a factory using those machines to give us high throughput sequencing in Britain for about £2,000 per sequence. And I'm on the advisory board of this organisation called Genomics England, which was set up after David Cameron pledged to take £100 million from their business development to put into this process. So what my challenge now with NHS England is to work out what do we do after that 100 million has been spent? How do we commission this? And it's not as straightforward as it might appear, because there are now 5 technologies out there that can do this, and if any one of those technologies was applied to any one of you, it would find about 3 million variants. Worryingly, they aren't all the same. So different machines give you slightly different variants because of their technology. But once we've ironed out that technological variation, we still have the serious problem. What the hell do you do with 3 million variants per patient? Imagine doing that in 9 minutes in a consultation. There's another problem. In addition to those error rates, you sometimes get gaps with this sort of technology. So you miss bits, because if it's very GC rich, which a lot of genes are, you might not get a good sequence of that part. And how do you know you didn't see something? So in other words, inadequate depth of coverage means you might reassure someone incorrectly. Another big problem, interestingly, is that when we actually sequence DNA using different techniques, we find that large chunks of our chromosomes have been replicated. And so in conditions like autism, we're now very interested in copy number variation. It's not that the gene's broken, it's just you've got too many copies of it. And that kind of unbalances the process. And obviously, if you're just looking at lots of little jigsaw pieces, you might miss the fact that you've got more than one copy of a particular segment, which might be important. What your mother had for breakfast when she was pregnant affects your genes, the Barker hypothesis. Our imprinted genes control our response to our environment dramatically. So, in fact, that's why birth weight is so important in predicting your response to things like hypertension and diabetes. Imprinting involves the methylation of your cytosines and changes of your histone bodies. It's a chemical tweaking of your genes, which you don't detect when you do whole genome sequencing. And then finally, your genes come in huge numbers of bits, like articles in women's magazines. You know, you get the page 2, and then you get a Tampax advert before you get to the next bit of the story. So your genes are just the same. They're scattered out over a huge distance, and they have to be assembled. And it turns out some genes, you can make hundreds of different versions of it. So whether or not a bit's important depends on how you assemble the gene. So splice variants, you can't see that when you do DNA sequencing. So I made this slide while I was sitting in Melbourne, passing an hour or two, because it struck me that in fact this isn't whole genome sequencing, it's whole genome sequencing. There's all sorts of problems that it doesn't detect, and we shouldn't overinterpret it. There is another problem. This is Jim Watson, who's actually the patron of our centre in Newcastle of Watson and Crick fame. And this is a few years ago when he came to visit. And this is his genome assembled by the people in Baylor. He had more than 1 million variants in and around his genes. And if you look at this top line there, missense variants, he had 8,500, nearly 9,000 known variants, and 2,500 we'd never seen before. And these are missenses. So the letter changes had not been completely synonymous. What it had done is it changed a proline to an arginine or a glycine. And so you're left with this question, what difference does it make having a proline or an arginine at position 29 in the gene? The gene still looks fine, it's just slightly different. And so we have this huge problem. And in fact, what we've got coming is the biggest traffic jam in history, which will overtake the existing world record holder, which you might be interested to know, was in China in 2010. It was 100 kilometers long and it lasted 11 days. So next time you're stuck in traffic, remember it could be worse. But we're heading for the biggest traffic jam in history. We've got to get bioinformaticians trained like never before to solve this problem. And so I'm going next week to San Diego. I'm now part of an international effort to do what's called the BRCA Challenge, which I put forward earlier this year to our new Global Alliance for Genomics and Health, which sounds rather grand, but this is a grand problem. These are 3 databases that exist currently in the world, one in Leiden that I'm involved with, one in France and one in America. And the blue dots show their overlap, or the Venn diagram shows their overlap. In other words, we've all got a pile of variants, but they're all different. And we haven't worked out which ones are real and which ones aren't, and got them all into one place. So we're going to try and create a global database so you can look it up and see immediately whether someone has a problem or not. And this is just an example of how that would work, because we started this earlier with a project called the Human Variome Project. And I helped to create a database which is now running globally in Leiden, which has all the mismatch repair gene defects on it. But this family came to see me in Newcastle, and Mum at the top there had Lynch syndrome. She had a positive family history of bowel cancer, she had a microsatellite unstable cancer, and she had an MLH1 leucine to arginine at position 559. And I didn't know what that meant. And on our database, it said variant of uncertain significance. And that's important because her daughter, who was there in the white coat with her family, didn't have it. So if it was the cause of the cancer, I could send them all home. They don't need a colonoscopy. But the books didn't tell me. Fortunately, we were having our Variome project meeting in Paris, and I was sitting next to Robert Hofstra and Rolf Simons, and they gave me all this bullshit here. You don't need to know what it means. I didn't really understand most of it. What they were telling me was that that is a pathogenic variant. They have described it. It's been written up. Therefore, I can reliably tell that lady that she's not going to get cancer because she doesn't have that pathogenic variant. That's why it's really important to get this sorted out. Now I'm going to get to the bit where I think you need to be very cautious of people like me impressing you. Because there's a sort of, you know when you go on those infinity pools when you go on holiday, and it looks like you can swim from here right out to sea. Well, that's what people do, is a bit of smoke and mirrors. So what I've shown you so far is the infinity pool. Now let's think about what about if you go out to sea. And most of our political leaders think like Dilbert's cartoonist who said, we ran a computer model on your DNA and your education, and we predict you'll die in a stapler mishap next week. That basically people think that because things are genetic, and you've all been to medical school, you've all fallen asleep to slides like that, haven't you? You know, basically, what the hell does that tell me? There's an 85% genetic contribution to schizophrenia. That's really interesting, isn't it? What are we having for tea? Basically, having your whole genome sequenced does not sort that out. And let me give you an example of that. And I'll take you back 150 years to Francis Galton, who had measured the height of all the parents coming to the Great Exhibition with their adult offspring and worked out there's a mathematical relationship. And that was turned into the polygenic Gaussian curve, the threshold trait, which I used to fall asleep to as a geneticist. So I want to use this discovery of the genetics of height to illustrate this concept to you. So this was when I was born. I was 6'2". I'm at the top end of that Gaussian curve. I'm no longer because everybody got a bit bigger. But that's the Gaussian curve in my age group for height. And you can see that it's a perfect curve. There's some people in the middle and some at either end. So that's because there's lots of genes determine your height. But let's imagine just for a moment, because it's easier at this early time of the day, to think there were only 2 genes that decided your height, and each of those genes only had 2 alleles. And those 2 alleles can either be heads or tails. So the gene on the right could be TT, and the gene on the left could be TT, and therefore that person at the extreme right has TT, TT, 4 Ts, very tall. If you've got 4 H's, you're very small. You can see, if you think about it, and you can do this in the bath, but you lose the coins if you're not careful, that there are actually 6 different ways of having 2 heads and 2 tails. So it's a lot easier to be in the middle of a Gaussian curve than it is to be at either end, because there's 6 different alternative versions of 2 heads and 2 tails. And there are 4 where you've got a 3 and a 1. So you've got a 1 to 4 to 6 to 4 to 1. It's a sort of early Egyptian pyramid. That's your Gaussian curve in its earliest form. Now imagine that we did a genome-wide association study for a condition which I've suffered from since adolescence, called bumping your head while walking under a door. And so the accident emergency doctors do a research project, and they do a genetic study of all the people who come in with frontal hematomas. And they find a gene for it. And they decide to write it up. It's called BYH1, bump your head 1. And they don't quite know why people bump their heads, but they know there's a gene for it, and it always tracks. And the red bits are the gene on the right. So if you're homozygous TT for that gene, you're much, much more likely to bump your head. In fact, it picks out all the very tall people who always bump their head, and it picks out 2 out of the 4 people who bump their head when they pass exams. The interesting thing is it also picks out one person in the middle who actually is never going to bump his head, but he's told he's going to bump his head, so he spends the rest of his life walking around worrying about bumping his head when he's never going to. Meanwhile, 2 of the 5 on the right are told by their GP, it's okay, you haven't got the gene. They rush out and bump their head. So, to make matters worse, a consortium of 100 people, I couldn't get all their names down from PowerPoint. 100 people did a paper in 2010, and there are 180 loci currently identified to determine how tall you are. But that's only a few % of the variance. We think it's about 600 of your 20,000 genes influence your height. That's not to mention whether you're male or female, whether your mother loved you when you were a little child, whether she ate the wrong things when she was pregnant, whether your ethnic background is a bit mixed, etc., etc., etc. You can see, and of course, this imprinting thing, we're all getting bigger anyway. So just something as simple as bumping your head, incredibly difficult to solve. I'm going to have to stop in a couple of minutes, but I just want to end by telling you one solution to this. Obviously, we've got to keep on going. We will crack this. We will find the pathways that cause schizophrenia and all these conditions by analysing their genetic panels, but don't be persuaded by 23andMe to pay a fortune to have your TT measured. It won't tell you anything. But one of the things you can do is focus on the things that you understand. And this is Charlie Lieber, the chemist in Harvard, who discovered nanowires. And he pointed out that if you charge a piece of silicon very, very thin, about the same diameter as a DNA molecule, and have something act on it, it will be detectable as an electrical event. And that so-called field effect transistor effect was picked up by my partner, Jonathan O'Halloran, who was working in the lab next door. And we now have the worldwide rights to nanowires to make DNA testing devices. And this is our little company called Quantum DX, based in Newcastle. And we've set up, there's about 50 of us now. And we basically invented, with the help of some Singapore engineers, little clusters of these 30 nanometer wide wires, and you can just stick a particular piece of DNA on, and if it binds, you can tell you whether it's A or B. We can do tests for simple genetic genotypes. And the thing is, this can be done very fast and very cheap. So our first cassettes will be used for warfarin, to predict your dose of warfarin. There are 3 SNPs I'm going to talk about in a moment. We're going to do a test for BRAF in tumours, which detects whether you'll respond to the new drugs. And we're, most importantly, perhaps going to produce, for Africa, a test for drug-resistant malaria based on the DNA of the malaria parasite. But just to talk about the one you're all involved in, warfarin. There are 2 genes involved in warfarin metabolism, the CYP2C9 and VQRC1, that contain very common variants in our population. There are 3 of them. And we've known for 15 years that if you test those 3 variants, you can predict people's dose accurately. It's not that exciting. It's like saying, don't buy shoes until you find out what size your feet are. But what do? We just give everybody the same dose and tell them to come back next week. Imagine if you bought your shoes that way. Try these 7, sir. Come back next week. We'll see if they're okay. So Munir Pir Mohammed here in Liverpool, along with the team in Newcastle and 1 or 2 others around Europe, have just published a fantastic paper on a huge series where they've proved the obvious. Genotyping people gets them on the right dose. You get fewer overshoots and you get them to the therapeutic window more quickly. And so we've now started to roll this out in Newcastle with our Academic Health Science Network just in time. You can't see the numbers here, but these are the practices in Newcastle, and the red line there is the number of people who are on warfarin, and the ones underneath are all the other NOACs, as you're all familiar with. And that's a 4-fold increase in our expenditure on oral anticoagulation for a 10% increase in patients. We can't afford to put everybody onto NOACs, and we don't need to. You all read the dabigatran stuff over the summer. Basically, the NOACs aren't all they're cracked up to be. They're about the same as warfarin, but that's warfarin without genotyping. And so what we're trying to do is demonstrate that warfarin can be kept being used if you genotype people. At the moment, we're using a pretty heavy piece of kit called the Sequenom, and that basically does your genotyping by measuring time of flight in our hospital. And that's going to be funded free in Newcastle initially. But then once we've proven that we can get this rolling with the primary care teams, we'll then switch to our machine. And this is our machine. It's not quite handheld yet, unless you'll be pretty strong, but it will get smaller. Basically, it takes these cassettes, and this cassette contains DNA extraction in 2 minutes, a PCR machine that you can throw away, and a nanowire chip that does that little test. And this is George Freeman, the Minister for Life Sciences, in case you didn't know who he was, and he's got an A level in biology, he tells me. He outqualifies the rest of the government. And so basically those cassettes will cost about 20 quid and give you a DNA result in about 20 minutes. And the machine will tell you what the dose should be. So we hope that that will help. And it takes closer to my final slide, Mr. Chairman, you'll be pleased to hear. This is our son, Jamie, in the year 2000, just finishing university, standing next to a horse chestnut in our backyard, which he planted when he was 4. And I asked him why he was planting the conker in the garden at the age of 4, and he said because he wanted to build a tree house. And I didn't have the heart to tell him it was going to take a while. And so in the same way, the Human Genome Project was launched in the late 80s, and in 2000 it was kind of finished, but it wasn't there yet. And so I used to go around showing this slide and saying, personalised medicine is coming, and so is Jamie's treehouse. Well, last summer, after some considerable discussion with our grandchildren, and we're against my wife's better judgment, we said it's time to build the treehouse. And so we built the treehouse. Well, to be precise, the carpenter John who lives across the street built the treehouse, but I put the roof on. So, ladies and gentlemen, identification of the monogenic causes of 1 in 17 of our population is affected by one of these rare diseases, and that's why you'll still need geneticists in the new world. And there are situations, such as in Lynch syndrome, where we can make a big difference. DNA sequencing is now cheap. We face variant inundation. Power to predict common disease is being grossly overestimated, but point-of-care testing might help us out with things like drug sensitivity, and if it does, I might get rich. Thank you for your attention.